Hi friends, if you like my videos, please subscribe my channel and also press the bell icon for the latest updates. Thank you.
Hi friends, my name is Fanindra. So in this video I am going to explain you about type 3 and type 4 hypersensitivity. Normally this video is a sequel of the previous video, whereas if you see in the previous video I explained you about type 1 and type 2 hypersensitivity, right? So in this video I am going to explain you about type 3 and type 4 hypersensitivity. So coming to the type 3 hypersensitivity, this is the type of reaction where aggregation of immune complex takes place which causes the damage to the tissue by complement activation. So here immune complex matlab immune complex is nothing but the antigen and the antibody together is said to be as immune complex, where everyone know that. So if you see here, this is the antigen, this is the antibody, right? Together it is said to be as immune complex. And here if the this is the type of reaction where the aggregation of immune complex takes place. So when the antigen and antibody will get binded over, which causes the damage to the tissue, and when this causes damage to the tissue, when this causes damage to the tissue by the attachment of this complement, when the complement will get attached to this immune complex, normally this is the immune complex, right? And when this black color one which I have drawn is known as complement system, and when this complement system or as a complement molecule will get attached to this immune complex, then what happens, the total tissue will get damaged. And that type of the type of sensitivity is said to be as type 3 hypersensitivity, and in brief I'm going to explain you now. So normally before entering into the phenomena, firstly you have to know what is meant by this complement system. So to know about this complement system, another big video I'm going to explain you later, but in this video I'm going to explain you shortly what is the complement system. And normally if you see in the complement system, when this complement system will get divided, then what happens is it mainly forms two components, I mean two segments, B segment and A segment. If you see here, this is the largest part and this is the smallest part, right? And the largest part is said to be as B complement and the smallest part is said to be as A complement or as A segment or as B segment. Okay, so now let us discuss about the phenomena of this type 3 hypersensitivity. So normally what they are given in the definition is that this is a type of reaction where the aggregation of immune complex takes place. That's nothing but the antigen, first the antigen will get will get attached to the antibody. So this is the antibody and this is the antigen. This both antigen and antibody will get attached with each other. Then what happens is that immediately signals will be responded in such a way that this complement system will enter into this blood vessel. So what happens is that when this complement system will enter into the blood vessel, then what happens is that the complement will get attached to the immune complex. There's nothing but immune complex is nothing but antigen antibody together as said to be as immune complex, immune complex. Then this complement will get attached to that immune complex. So what happens when this complement will get attached to the immune complex, I have said you here. So what happens is that the complement system will get broken down. So when this broken down takes place within this complement system, then what happens, complement B and complement A will get will get divided. So if you see here, this is a complement B and this is complement A. Normally complement A will get passed out from the blood vessel. It will move out from the blood vessel in the form of toxin, and this is very harmful reagent. And next, if you keep this aside, if you keep this component A, I mean or else fragment A aside, then another fragment B will be present over here, right? And that fragment B is very much responsible, takes place within this type 3 hypersensitivity. And if you see here, this complement B or else this fragment B will release us or else will helps to release us these interleukins and histamine chemical agents. So these are the very harmful chemical agents. And if you see this interleukins are of various types: IL-6, IL-7 and IL-2. And here IL-2 interleukin is very much harmful, and that IL-2 also releases perforates and granzymes, and there is something left over here, and there is not very much required for you, but this interleukin is very harmful. That only the one thing you have to remember. And this interleukin and histamine are very much dangerous. So what happens is that this is the blood vessel, right? And total process takes place in the blood vessel only. Okay, and if you see the blood vessel, normally we all of us know that the blood vessel consists of layers, right? I mean epithelium and endothelium layers. All of us know that, we are learning from this since childhood. We are learning that normally blood vessel consists of layers, epithelium and endothelium. So these are the epithelium and endothelium. Normally epithelium and endothelium layers will get combined with each other if you observe through a microscope. Then after this releasing of these enzymes, or else if you see these chemical agents from the complement B, then what happens, if these blood vessel layers are exposed to these layers, then what happens is that gaps junctions will be formed. So that's nothing but the gaps will be formed between each of the each of the cell. So then what happens, the total layer will get damaged. So that's nothing but the total layer will get damaged, and that's nothing but it becomes very thin. So what happens, as the blood vessel will get becomes damaged, then what happens, automatically the tissue will also get damaged. So now you can understand the definition. If you see here, this is a type of reaction where aggregation of immune complex takes place which causes damage to the tissue. So what happens when this antigen antibody will get combined over here, complement will get attached. In the same way, the complement B and complement A will get divided, and the complement A will passed out from in the form of anaphylatoxin, which is very much dangerous. And if you keep this aside, the complement B will releases or else will activate the enzymes or else the chemical agents like interleukins and histamines. As these are very much dangerous, it mainly helps in thinning the layer of this blood vessel layer. I mean, the gaps will be formed in such a way that the total blood vessel will get damaged. As this total blood vessel get damaged, then what happens, as this normal blood will passes throughout the body, then what happens is that the total tissue will also get damaged. In this way, the type 3 hypersensitivity phenomena takes place. So now let us discuss about the type 4 hypersensitivity. So this is very much important and very much if you see here, this is type 4 hypersensitivity, and we can define it as, it is a delayed hypersensitivity. It is a delayed hypersensitivity which is mainly mediated by T helper cells. T helper cells. So now let us see the phenomena. It is very much easy and very much important also to remember. If you see here, listen properly. Normally, this type 4 hypersensitivity is mainly caused or is mainly attacked for the person when he is exposed to a pollution area, or else if he touch a poisonous plant. If he touches a poisonous plant, in this case we take an example of, if we touch a poisonous plant, then what happens is that normally that poisonous plant consists of haptens, right? And haptens is a type of antigen. If you think this is a haptens, if he touch that poisonous plant, then haptens will get enter into his body. So when this haptens will enter into the body, then what happens is that this hapten and normally carrier proteins are mainly present, carrier proteins are mainly present in our body. This hapten and carrier proteins are combined with each other to form peptides. So haptens will be released from the plant and the carrier proteins will be present in our body itself. So both that hapten and carrier proteins will get combined over with each other and mainly form peptides. So this is the structure of the peptide. If you see this hapten and carrier protein will get combined to form peptides, and now what happens is this peptides will enter into the macrophage. So here normally, if you see in the case of immunology, if you see in the way of immunology, this is said to be as antigen presenting cell, APC. As the best examples of antigen presenting cells are macrophages, dendrites, and leukocytes, right? You know about leukocytes, right? White blood cells. Leukocytes are nothing but white blood cells. So I am taking in this case as macrophages. So normally it is said to be as antigen presenting cell only, but the best example of antigen presenting cell is macrophage only, right? So when these peptides will enter into the macrophage, then what happens is that normally this macrophage consists of vesicles like this. It consists of vesicles, and within that vesicles, what are present? Lysozyme is an enzyme which is present over here. This black color dots which I have written over here is lysozyme. Wait a second. So if you see here, this black color one which I have drawn here are known as lysozyme. So what happens here is that peptides which have entered into this macrophage will get combined with this lysozyme enzyme. So then what happens is that lysozyme enzyme will break these peptide linkages. I mean, that's nothing but when the peptide enters into the vesicles, then this lysozyme will break down that peptides. So due to that breakage of that peptides, or else due to the chopping of that peptides, what happens is that all of this may release to the will come out of the vesicles. And as we know that there are many vesicles which are present in the macrophages, and within another vesicles, MHC class 2 molecules are present. If you see here, these are another vesicles. All of these are vesicles only. In another vesicles, MHC class 2 molecules are present. And what is the main function of this MHC class 2 molecules? MHC class 2 molecules will attract the fine divided particles from these vesicles. So normally, if you see here, this lysozyme will break down these peptide linkages, right? And when these peptide molecules will get break down, then all of these small fragments will be released out, and that fragments are normally said to be as antigens. So you should not say it as antigens, you should name it as allergens, so that all of these allergens will get attracted towards this MSC class 2 molecules which are present in the another vesicles, as they are present in the other vesicles, right? So what happens is that when this will attract all of these fragments which has been released out from this vesicle, then what happens is that this total vesicle along with this class 2 molecule will be exposed towards the membrane. will be exposed towards the membrane. Then like this, if you see here in the case of this diagram, it will be exposed towards the surface of the antigen presenting cell. That's nothing but the macrophage. Then what happens now? Now what happens, as before only I have said you that it will be mediated with the help of T helper cells, right? So now T helper cell takes part. If you see this is the T helper cell, and this T helper cell will get to know that there are antigens which has been coming out from the antigen presenting cell. As the name itself, it indicates antigen presenting cell is nothing but the cell which represents the antigen at the surface are known as antigen presenting cells, where we all know that. And now these T helper cells will we all of us know that these T helper cells normally consist of T cell receptors. So these are the T cell receptors, and these T cell receptors will get binded over with these MHC class 2 molecules. So when these T cell receptors will get binded over with this MHC class 2 molecules, wait a second, these are known as TCR, T cell receptors. Okay, so when these T cell receptors will get binded over with this MSC class 2 molecules, then what happens, as antigens, I mean, which I have said you previously, and all of these are allergens. We have to not call it as antigens, you have to call it as allergens. All of these antigens or as allergens will enter towards this T cell receptors and finally enters into the T helper cells. And then now what happens, when this enters into T helper cells, then this T helper cells has a capacity to produce interleukins, chemical agents like interleukins. As we all of us know that before only I have said you in the type 3 hypersensitivity, interleukins are very much harmful. If you see here, interleukins are very much harmful, and there are types of interleukins I have said you, right? Interleukin 6, interleukin 8, interleukin 2, and among these all, interleukin 2 plays a major role. Normally interleukin 2 is very much dangerous when compared to this IL-6 and IL-8, I mean interleukin 6 and interleukin 8. Of course all of these both are also dangerous, but when comparing to this is very much dangerous when compared to this. Okay, so normally this IL-2 has a capacity to release enzymes or as the chemical agents like perforins and granzymes. So now what these perforins and granzymes does, normally these perforins and granzymes will kill the macrophages, so in this way the antigen presenting cell will get totally killed. So normally what happens, these T cell receptors will get binded over with the MSC class 2 molecules, then the antigens or else normally these antigens or else allergens will enter into the T helper cells. When this enter into the T helper cells, then what happens, this TH1 cells or the T helper cells will get recognized that antigens are coming into it. Then what happens, immediately it releases IL-6, I mean interleukin enzymes or else interleukin chemical agents. And here among these three, or IL-2 or else interleukin 2 is very much dangerous, and this will release further perforins and granzymes. As both of these are very dangerous, then what happens, it helps in the killing of these macrophages or as the antigen presenting cells. As these macrophages are present in the tissue, what happens, the total tissue will also get damaged. In this way the damage of the tissue takes place, and the damage is said to be as type 4 hypersensitivity. Okay, and this is up to a part. And if you see another part, if again another carrier protein will bind with heptane and will enter again for the second time. Normally for the first time what happens, the macrophage will get killed by utilizing by these enzymes or all as the cycle which I explained you now. The total macrophage will get killed, right? And again this happens for the first time, and what happens for the second time? So again the heptane molecules or this will get binded over with each other and forms peptides, and when these peptides will enter into the macrophages and the total process begins same as it is, then what happens here is that again T cell receptors how these T cell receptors could recognize that the same antigen has been again arrived from the from the host. How it recognizes? It recognizes because of the memory cells. Normally T helper cells consist of memory cells. Okay, T helper cells consist of memory cells. This is very much important to remember. Due to the presence of these memory cells, what happens is that it can recognize the type of antigen which has been repeated same, and if it is repeated same, then again the damage of tissue will not take place. Okay, so in this way this is very much important to remember type 4 hypersensitivity. And for proper notes of this total hypersensitivity, we have a group in WhatsApp, and the link of that group, invite link, will be given in the description box. So if you people are interested to take notes from me, so if you message there, then immediately I will give you notes there. Okay, PDF, I will send you PDF. So thank you for watching this video, guys. If you like this video, please do like and subscribe, and if you have any doubts regarding this video, please comment in the comment box, or else you can ask also in the group also. Thank you.